Okay, so I'm guessing there'll be about 3 times as many people here on Tuesday at 8 a.m. than there are now. I hope there will be. That would be good. So basically you get to ask questions for all of them who aren't here. Yes? I probably should have said I was going to do clicker questions, and then there would have been a lot more people here. So be it. But we don't have time for that. We only had 10 weeks to cover all of virology, which is not enough time. Exactly. Yes, I know. I should do a whole year of this, but so be it. I always promise to just use the same slides that I have before for my reviews, and I always break my promises. This is what we talked about since the last midterm. The final is not cumulative per se, but I will expect that you at least remember some of what we talked about before the last 2 midterms. So we really focused on the DNA viruses, the, well, really tiny ones, the single-stranded DNA viruses, the polyoma and papillomavirus getting bigger, adenovirus is bigger yet again, herpes virus is bigger yet again, pox virus is bigger yet again, and then the megavirales even bigger, just in terms of their virions, but also in terms of their genomes. Then, because we can't not talk about them, we talked about the retroviruses, and then, of course, the most important viruses on the planet, like my friend up here, SSV1, the archaeoviruses. So as usual, stop me, ask questions as we go. There is absolutely no guarantee that the stuff is on the review will be on the final, because I write questions on pretty much every single slide that we have done, and then pick and choose from those afterwards. Yeah?
Why did you not include the retroviruses and archaeoviruses and like this is bigger than this, it's bigger than this?
In terms of the sizes of virions, it's more of a progression issue. But as we all remember from last lecture, right, on the archaeoviruses, there's some pretty small ones and some pretty big ones. So it's a much larger group. The retroviruses is actually a pretty constrained group of viruses. But if you remember, I'm sure you do, from the archaeoviruses, there was one which is single-stranded DNA, as well as double-stranded DNA, and that one's got the largest of these single-stranded DNA genomes. But yeah, these are sort of, how you organize this course is a really kind of open question. You'll notice the textbook mostly went through in this order, and then we jumped around a little bit. Okay, so again, some of the highlights, lowlights, probably depends on what your point of view is of these various different virus families, or in the case of archaeoviruses, massive group of families. The polyoma or papovaviruses, as people used to call them, but also the papillomaviruses. And we didn't sort of emphasize this, I think, potentially as much as it should have been. All of these are packaging their genomes in nucleosomes. So cellular packaging process, and also, not surprisingly, almost completely cellular processes, which are involved with these, not only packaging, but also in terms of replication and transcription. In terms of SV40, this is a genome that, again, should look a lot like lambda from that, again, stuff that we've covered in previous parts of the course. You've got divergent promoters around a central regulatory region. This central regulatory region is also the origin of replication. This is where you have binding of lots of different proteins, mostly cellular proteins, but then the answer to all SV40 questions? Large T antigen, exactly, which also is involved in not only transcriptional regulation in terms of stimulation of these early genes, repression of late genes, but also for replication. So again, large T antigen is what's doing most of these things. One thing that I neglected to mention, I think, the first time through is this part of the genome down here at the bottom where the transcripts end up running into each other. This is what happens if you have circular genomes. when you have transcripts that are running into each other, you're going to end up with double-stranded RNA. And that double-stranded RNA turns out to be something that many cells are really good at in terms of recognizing that there's a viral infection. And so that's one of the aspects. We already talked about the RNA viruses, again, way back when, the earlier part of the course. You end up with double-stranded RNA because you have to have that as part of the intermediate of your replication process, but also for DNA viruses, you very often have double-stranded RNA because you've got lots of these open reading frames, transcripts, which are going to overlap with each other. Large T antigen, again, we already talked about as being a transcriptional regulator and a replication protein, it's the helicase, but this also is interacting with all of the proteins that are important for the cell cycle. So all of these DNA viruses dependent, with the exception of the nucleocytoplasmic large DNA viruses, on cellular replication processes. So they've got to stimulate the cell cycle somehow. And that's true for these, the papilloma, the polyomas, the adenos, the herpes viruses as well, to some extent, a little less so for the herpes viruses, to stimulate the cell cycle. So interacting with RB that normally is repressing the cell cycle, nuclear localization signal because it's got to get itself into the nucleus, and then P53 in terms of interactions with, again, cell cycle regulatory proteins. And the P300, oh, sorry, Nicole.
No, you're fine. So, like with E6 and E7,
yeah.
papilloma, is large T antigen degrading these proteins, or is it just binding to them and keeping them from doing what they normally do?
Okay, so the question is basically, what's the mechanism of large T antigen versus what we see with E6 and E7? And to some extent, you probably also mean E1A and E1B for those proteins. We'll get to that in just a second. So if I can, make a little pause on that question, that would be best. And then P300 is just a reminder again that this is a transcriptional regulator. P300 is a co-activator in terms of transcription, and that co-activator is now very well known for dissociating nucleosomes. Again, not surprising because it's the nucleosome template. It's what you have in these papilloma and polyoma viruses. The papilloma viruses basically have taken the large T antigen and split it into a bunch of different proteins because one protein, you don't have all the activity in one protein. Well, it's evolution. There are two different ways to solve the same issue. So here we have E1 and E2, which are the proteins that are mostly involved in the transcriptional regulation and replication, and E6 and E7, which are mostly involved in the regulation of the cell cycle. And so this is where I wanted to get to before answering your question, Nicole, is looking at these various different proteins. So human papillomavirus, E7, interacts with RB, and the interaction with RB is literally just binding to RB so that RB can't interact with the cellular E2 factors. And so normally, again, it's just protein-protein interactions. RB interacts with these E2 factors and represses their activity, and in this case, we're talking human papillomavirus, it's E7 that interacts in the same way. SV40, exactly the same deal. So interacts with, pulls it off. E1A also seems to act in exactly the same way. Now, the big question here is, of course, because large T antigen is interacting both with RB and P53. And so E1B is involved in shutting down apoptosis, because that's one of the things that P53 does. And through that process, the large T antigen just seems to hold P53 so it's not active, whereas E6 seems to lead to degradation of P53. So there are multiple different ways of dealing with P53, but they're slightly different processes. Yes, no, huh? Okay, that's it. There will be 6 or 7 questions on papilloma and polyoma viruses, which is true for every single one of these sets of viruses that will be on the exam. So next one are the adenoviruses. These are really fascinating structures. Now, I think they're beautiful, but that's a different story. You may or may not believe that. It's in the eye of the beholder. But here we have multiple different proteins that come together to make a otherwise what looks pretty icosahedrally symmetric particle. And it's really hard to ask good questions on structures. Trust me, I try. So these have, again, these multiple different proteins. We've got the pentons, bases, penton fibers, and the penton knobs. This, of course, is what's interacting with the primary receptor, the adenovirus receptor, and then mostly hexons, which we have here between each of those pentons. You can't see here that it's t equals 25, but here it should be pretty obvious. 1, 2, 3, 4, 5. From one 5-fold axis to the next 5-fold axis of symmetry. Of course, it's a pseudo t equals 25 because you've got lots of different proteins that are involved in this process. And you have all of these, I like to call them interstitial proteins. I'm not going to ask you all the names for them because I can't remember all of these names. Pentons and hexons would be good to remember. 8, what's that, 6N, 3A? Don't bother. And again, these are these minor proteins that are helping to hold this whole process together, and there are lots of those minor proteins. So that's the structure of the adenoviruses. What's inside the structure is this fascinating genome that we learned all about splicing by studying the genome of adenovirus. We learned a lot about transcriptional regulation by studying adenovirus, and quite a bit about splicing as well, sorry, not splicing, tailing as well by looking at this process. And the take-home messages here, we've got early proteins, E1A and E1B, that we already talked about, those ones interacting with the cell cycle. The other early proteins are going to be those that are involved in DNA replication processes, DNA binding proteins, the terminal protein, because this is protein-primed replication, the DNA polymerase itself. These E2 proteins are the ones that are being positively regulated by the E2Fs. Now, E2F, again, this is one of those bizarre, again, because people learned about the cell cycle and transcriptional regulation by studying adenovirus. E2Fs are cellular transcription factors. They're not viral factors. So those E2s are all, and they are important for, surprise, surprise, expression of these E2 genes in adenovirus. Then we have these smaller pieces, and then, of course, the major late promoter with this tripartite leader sequence. This is what gets spliced onto the 5' end of all of these late messenger RNAs, 5 different classes, depending on where each of the poly A tails are being added. And each of these, pull separate classes, and then differentially spliced like mad. Small RNAs, people didn't think that small RNAs were important early on in terms of regulatory processes, but the adenovirus guys said, of course they're important. These small RNAs here turn out to block the PKR effect. So what the heck is PKR? We talked really briefly about PKR before. That's protein kinase R. Protein kinase R recognizes double-stranded RNA. And when it recognizes double-stranded RNA, it says, hey, cell, watch out. We've been infected by a virus. It's time to shut down the only thing that no virus has, which is translation. So shut down translation through the activity of PKR. And you remember those double-stranded RNAs that you see at the end of the polyomaviruses. These will also be stimulating PKR. These two small RNAs actually bind to PKR and stop it from having that antiviral effect. So as usual, when you think about cellular defense mechanisms and viral infection mechanisms, these are always evolving in sort of what a lot of people call an arms race. You have viral defense mechanism, the virus gets away from that defense mechanism, the cell comes up with a new defense mechanism, and so on and so forth. And so this is a nice example of that. It's the small RNAs in adenovirus. Adenovirus replication, as I mentioned already, is quite different because we have protein-primed replication. Protein-primed replication due to our pre-terminal protein, which is one of those E2 proteins, provides a OH, which a cytosine gets attached to. The identity of this nucleotide is not important. Don't bother. But it has a nucleotide that is attached to a OH. That will then provide your 3' OH. The viral polymerase will find this 3' OH, has a template, will replicate its way down one strand of the genome, and that single-stranded DNA binding protein, which we also saw as one of those E2 proteins, binds to the single strand and basically serves as the helicase, pulls those two strands apart. When you get down to the other end of the genome, the ends of these genome, because they have inverted terminal repeats, mean that this sequence, if it didn't have a typo like it did in the book, will also base pair with this end of the genome. So you can make these, they call them in the book the frying pan structure. And so this can also serve as a template for the DNA polymerase to make an extra copy of the genome. Happy with adenoviruses? Questions on adenoviruses? No? Yes? Good? Cranking through this stuff. Cool. Herpesviruses. This is, to be perfectly honest, probably the lecture where I feel least confident about my knowledge because I've had to guest lecture, do it all the time in the past. So if I get something wrong, point it out to me, and I will try and correct myself. But here, the main things that we talked about are going to be herpes simplex virus 1 and 2, which in fact are extremely similar at a molecular level. So similar at the molecular level, who cares about what kind of diseases they cause? And then varicella zoster virus, which is the chicken, pox again, just because it causes these kinds of lesions. But chickenpox is a herpes virus, and one of the major problems with all of these herpes viruses is that they can go into a latency, which conceptually is really similar to what happens with lambda, but in terms of the actual regulatory mechanisms is completely different. But conceptually, hangs out for a while in the nucleus until conditions get bad, some kind of stress condition, and then will be reactivated. So that's the whole idea of the latency, and extremely active area of research, meaning we really don't know very well how it happens, is that process of reactivation and how reactivation happens. These HSV, of course, cold sores, et cetera, and again, it's exactly that same kind of latent induction of virus production that happens. Cytomegalovirus that I really didn't have much of a chance to talk about, but if you're interested in more about cytomegalovirus, check out a whole bunch of stuff which is going on at the Vaccine and Gene Therapy Institute on the West Campus of OHSU. They're probably the world's experts on cytomegalovirus, and certainly in terms of cytomegalovirus and applications for vaccine development. So if we look at the HSV1 genome, we also talked about the fact that all of these herpes viruses have really funky ends to their genomes, and lots of recombination, lots of repeated sequences, probably mostly important for packaging purposes. HSV1 has 2 unique segments, large one and a small one. Gee, thanks. Can't you come up with a better name for these things? And then repeated sequences on either side of them. Anytime you have a prime, that means it's the complement of this sequence. So we have direct repeat at either end, and then these inverted repeats that are present here in the middle. And if you just purify virions, purify genomes from each of these herpes viruses, you find a mixture of all of these things flipped back and forth relative to each other. And again, that's probably due to the rolling circle kind of replication that happens late in infection in the packaging process. Yeah?
Are we still doing that A sub n is defined as A? Same thing.
So here, I think this is, I would say this is a typo. Okay. So these are identical sequences. A, N, and A. Those are identical sequences. A prime is the complement of that sequence. So it's just be flipped around. Okay, these are not linear genomes when they're inside the cell. They're circularized, and so if you look at a circular genome here we have these repeated sequences, again, B and B prime, and they're complements of each other. It just means that on the opposite strand. And so if you look at this transcript here, this latency transcript, it's going in this direction, latency transcript going in that direction. But these are identical sequences just on both strands here, and opposite orientations relative to each other. And that means that any of the genes that you have present in these parts of the genome are present in multiple copies, and we'll look at what they do in just a second, particularly alpha 0 and alpha 4, also known as ICP 0 and ICP 4. And then some of these other proteins, again, early on in the replication process. Yeah, Jacob.
Do you have alpha, beta, gamma types of herpesviruses and alpha, beta, gamma types of proteins in HSV1?
So yeah, the question is, again, just for people who didn't hear in the back, do you have alpha, beta, and gamma proteins and alpha, beta, and gamma herpes viruses? The answer is yes. And I will try not to be too confusing about those different things. You know, what kinds of, is cytomegalovirus a alpha, beta, gamma, delta, epsilon herpes virus? I don't think that's a really reasonable question to ask. And I will try and stick with one nomenclature system. And we've got, UL, et cetera, UL41, UL48. I will try and always call these one thing. If I've got something else that's in one of the questions in one of the exams, say, Dr. Stedman, is there a different name for this? Feel free to ask me, but I'll try and be consistent about these things. So yeah, what the heck is UL21? I have no idea. So I can't follow all the names of these genes either. But conveniently, I put circles and arrows on the ones that I think are important against the ones that we've mostly talked about. So VP16, this is that transcriptional activator. And a nice reminder, someone who will remain nameless asked me some questions about VP16. And I posted the answers to all those questions under the discussions and questions part in D2L. So if you have more, and anyone else ask me questions, again, email-wise, I will try and answer them and copy those answers into the questions section in D2L. So VP16, this amazing transcriptional activator, but it doesn't bind to DNA by itself. It only can associate with DNA and have its transcriptional activation activity in the presence of host DNA binding proteins. So it's ubiquitous host DNA binding proteins that VP16 interacts with and then will activate transcription. Molecular biologists have taken advantage of this and done protein fusions where they have a DNA binding domain attached to VP16. And that turns out to be a really strong transcriptional activator dependent on whatever that DNA binding site is. But that's a completely artificial construct. Yeah.
VP16 has nucleosome displacement activity. Is that only in recombinant situations where, because there's no, is VP16 just in wild type herpesvirus going to be displacing nucleosomes?
So VP16, the activities of VP16, that's one of the neat things that people showed. Sorry, your question is, does VP16 just have nucleosome displacing activity in a herpes virus infection or just in general in some of these recombinant cases as well? And the answer is VP16 has all of these activities independently of where it's associated. So if you've made a recombinant VP16 bound to your favorite DNA binding domain, and in the case of most of the yeast biologists, it's Gal4, then it will do everything that VP16 otherwise would. So help recruit various members of the pre-initiation complex. Also, and the nucleosome displacement activity is also recruiting some of the histone acetyltransferases and the chromatin remodeling complexes that will then allow you to get rid of nucleosomes. So it's functioning in the same way in all of these systems. Which is one of the neat things about molecular biology is these things actually do seem to work similarly in other systems. So you can do these recombinant things and make sense out of them.
Does it show that in herpes wild type? Would we have known without recombinant techniques that displaces nucleosomes because it's not going to be affecting? Is herpes infection affecting nucleosome displacement?
Oh, so basically, okay, so I'm going to try and paraphrase your question. But is VP16 in a herpes virus infection doing the same thing as it does in one of these recombinant systems in terms of displacing nucleosomes and activating transcription? Yes, definitely. It's doing the same thing in both of those systems. Okay. And we can actually look at that here as well. So boxes, again, boxes and arrows are your friends as thinking about, again, next Tuesday. ICP0, that would have been the alpha 0 gene that encodes ICP0, ICP4, alpha 4, etc. ICP0 is one of the immediate early regulatory proteins, which again, immediate early means it's going to probably regulate early proteins. Those early proteins are probably going to regulate late proteins. Makes lots of sense. ICP4 is kind of the odd man out a little bit here because not only does it activate early and late proteins, but it also is a negative regulator of its own production. And so transcriptional regulators, we already talked about this for lambda C1, can activate transcription and it can repress transcription, but unlike C1, you kind of turn it on its head. C1 usually stimulates its own expression. Here, ICP4 is repressing its own. Yeah?
TXN stands for toxin or activating, is that TXN?
Oh, TXN. Here, TXN is almost always going to be transcription. No, we have transcription here somewhere? Oh, here, activating TXN factor. That was Dr. Estes. No, Estep who wrote that, not me. So I will try and I'll try and write all these things out. So if I want an exam, I'll definitely not try and do TXN. That would be transcription, yeah. So it's a transcription factor, yeah.
Does ICP4 only downregulate? Is ICP4 the only protein that it's downregulating in immediate early, or is it also downregulating other immediate early genes that are not ICP4?
Good question. So the question is ICP4 also downregulating other immediate early genes? And the answer is, I don't know. And so if I don't know, it's probably not going to be on an exam. It's quite possible that it does. And how I would find out is I would go and look for ICP4 binding sites in various different promoters. But since I don't know if they're there, but that would be a relatively easy. I'm sure it's known. Again, it's one of these things that I don't know, but I know that it's known. It's like known knowns and known unknowns and all of these fun kinds of things. So ICP0 and ICP4, again, these are these transcriptional regulators. The other, probably most important of these is ICP27. Why is ICP27 so important? Because it's regulating splicing. And why regulating splicing? Look at all of these beta and gamma genes. Do you see many splicings happening in them? No. And what that means is all the splicing machinery can be blocked. You'll be making all of the herpes virus RNAs, but you won't be making cellular ones because you've blocked splicing. The mRNA control mechanisms are going to keep all of those messenger RNAs in the nucleus, so they won't get exported. So ICP27 and ICP22, as everyone was chatting about here earlier before class, is involved in regulating the cell cycle because just like all of those other DNA viruses that we've talked about, this herpes virus also needs to regulate the cell cycle in terms of turning on a number of genes, but a lot fewer because herpes viruses actually encode many of the proteins that they need, thymidine kinases, ribonucleotide reductases, etc., in terms of getting sufficient precursors in order to be able to make their genomes. Yes?
So this is the alpha protein in alpha herpes virus?
Okay, so as far as alpha herpes viruses, beta herpes viruses, gamma herpes viruses, forget that.
Okay.
Don't even worry about thinking about it.
Beta and gamma virus gene expression, but it doesn't say alpha. And so
Yeah.
alpha at the top.
So as far as we're concerned, and again, this is, don't tell any of the herpes virologists at OHSU about this, but when we say alpha, beta, and gamma, I would just take those as being immediate, early, early, and late.
Okay.
Good. That works. Okay. So herpes viruses, as I've probably beat on maybe too hard, are extremely similar in lots of different ways to bacterial viruses, particularly the bacteriophage. One of those aspects is the packaging and how we have replication. You have a rolling circle replication, which gives you concatenameric genomes, and this is probably where you have this recombination which is taking place between the different ends of the genome. And then these repeated sequences, so A, and this is probably where the A to the N comes from because you've got so many of them throughout these concatenameric genomes. These A sequences are packaged in apparently a specific mechanism where you end up with 2 copies of A at the end of your genome inside one of the capsids. This then is made into a separate capsid, and then the big difference relative to all of those bacteriophage, now it gets a whole bunch of extra proteins around the outside, which is called what? The tegument, and then a membrane around the outside of that. The tegument is very important because it also carries proteins that are important for really early steps in the replication of the virus, like what? VP16, exactly, which is going to be regulating right here, turning on these very first immediate early genes. Yeah?
Besides VP16, are there any other proteins or mRNAs that we need to worry about in the tegument that are packaged in VHS?
Yeah, virus host shutoff protein.
Just VP16 and VHS that we need to worry about.
Those would be the only ones that I would worry about if I were studying for the exam.
We need to worry about the number of proteins and the number of mRNAs in the tegument.
How many are there?
10 to 14.
Is that known for all herpes viruses?
No, it's just on one of your slides.
And so therefore you have to remember the numbers because Stedman really likes numbers because it's really easy to come up with wrong answers. But in this case, no, I would not try and remember that because it is actually really quite variable. And it does seem it's not the thing with tegument, so maybe back up a little bit. Yeah, I'm kind of making jokes about this in terms of what will be on the exam. But it is important in terms of how these proteins are associated, how you end up with these proteins and these RNAs associated with the virion. So are these direct specific interactions? Is there something that sticks off the capsid that says, hey, VP16 has to be associated right here? These actually seem to be pretty random processes, which is kind of interesting. So how do you end up with 10 or 14 of these RNAs always being packaged? Because there's no sort of obvious way that you can package these things around a capsid in a very specific way. So that the whole process of packaging of herpes viruses, packaging also for that matter of the pox viruses is a really interesting question that nobody has a really good answer to exactly how they work.
So VP16 is UL49, right? Not UL48.
Don't bother. VP16 is VP16.
Okay.
How many more questions can we let Jacob ask?
I saw asking questions a month ago.
Yeah.
Is there one of the bacteriophages that the packaging is more similar to, or the most similar to, or is it just bacteriophage? You can ask like T4 versus T9 or T4 versus T7.
I think that would be kind of up to you to think about what you remember about the packaging of these various different viruses.
So can I ask that question again? Will you give me an answer?
No, I'll tell you to go and review those bacteriophages. Okay, that was easy. Well, no, relatively speaking. There are some that make a lot more sense, and if you go and look at some of the old exams, you'll notice there are some questions very similar to that. There will be some pretty obvious ones, and it was true for all of these exams. There's one answer which is the best answer for any of them. Pardon? It better not be lambda. Nothing is lambda. No, that lambda is always like SP40 large T antigen. SP40 large T antigen. Again, this is also, again, thinking about coming up with the wrong answers for questions, you always throw up lambda, always throw up SV40 large T antigen. So people will think that might be the right thing. So I'm giving away all my secrets in terms of how I write exams. So that's why you come to this, right? So wanted to at least briefly cover, and actually probably spend a little bit more time talking about George's lecture, which I thought was awesome, by the way. Thought he did a wonderful job. Talking about these single-stranded DNA viruses that are infecting eukaryotes. Of course, we talked about FIX174 way back at the beginning of the course. Turns out that FIX174 in many ways is extremely similar to a lot of the circoviruses in terms of its replication processes. These are very small, the parvoviruses, the porviruses, T = 1 icosahedra, nice regular structures made up of 60 subunits, and of course either an H or a K of 1. Of the parvoviruses, these are different from FIX174 and from the circoviruses and the cruciviruses in that they are linear genomes. They actually have ends as opposed to being circular. And we'll look at that in just a second. George also mentioned permissive versus susceptible cells. And I didn't want to say permissive versus non-permissive. It's kind of bizarre that the textbook brings this up now. It should have been something that came a lot earlier. But the message here is that any cell which has the appropriate receptor is a susceptible cell. Doesn't mean it's going to be able to reproduce virions. But if it's got the right receptor, it's susceptible. If it's got the right cellular machinery, it will be permissive. And this is, again, probably why they mention this in this particular chapter in the textbook, is because these parvoviruses have practically nothing that they can use of themselves. They're incredibly dependent on everything else, again, partly why some of them are called dependoviruses. So to have the correct machinery, in the case of one of these dependoviruses, having that correct machinery actually means you're infected by another virus. To have the necessary machinery for replication. So in order to have a cell which can be a productive host, it has to be both susceptible and permissive. So have extracellular receptor, have all the machinery that's necessary for the virus to replicate. Again, above and beyond, of course, what the virus brings with it. Yeah?
So you're saying they need a helper virus?
Well, in the case of these particular viruses, they do need a helper virus. But just this is a general picture. So if you think about the, what we just talked about right at the beginning today, the polyomaviruses, those are dependent on having all of the appropriate DNA replication machinery, etc. When we're just about to talk about the pox viruses next, those actually require very, very little host machinery. And so that's the really sort of the difference there. And so you'll have some which are going to not be able to replicate in very many cells because they're dependent on so much on the host machinery. And the extreme is going to be these dependoviruses. It has to be another virus infection as well. Yeah, Nicole.
So for polyoma, you were, I watched previous years.
Mm-hmm.
But so if they're permissive, then it has all the machinery. So is that the same as this? Because those ones, are they tumor viruses? Or what would be the difference between this situation and the polyomaviruses and papillomaviruses?
Okay, so so the basic question is what's going to be a difference between a permissive cell for instance, one of these adeno-associated viruses or for SV40. So that's just another way of expressing it.
Susceptible wasn't brought up in that. It was just permissive.
Right, but so susceptible is just having the receptor.
Okay.
So just having the receptor. But permissive, and it's going back, so it's a nice review. And so if you think about SV40, particularly in just thinking about the replication process. The replication process for SV40, you have to have DNA replication. How does DNA replication work? You have to have primases, you have to have helicases, you have to have DNA polymerases, etc. Large T antigen is a helicase.
Yes.
So you don't have to have a helicase. So you can have a permissive cell for SV40 that doesn't have a helicase. In the case of one of these adeno-associated viruses, they don't. So they have to have a cellular helicase. And in these cases, they also don't have an SV40 to stimulate the cell cycle. So they also are going to require those proteins. And this is the ones that come from that extra virus infection, which are going to stimulate the cell cycle. So any permissive cell, if you've got a really, really small viral genome, you're going to have a lot of machinery inside the cell. If you've got a really big genome that carries a lot of machinery with it, then the actual cell can be permissive. So what that means is lots more cell types are going to be permissive for these really big viruses than there are for the really small viruses. Does that make more sense? Okay, good. Yeah, this is a funky concept, and again, we should probably have this the beginning of the term rather than right here. But that's the way the textbook puts it together. So, so do they need anything? Yes, they do still need a few things in terms of their viral genomes in order to be able to replicate. Of course, they need structural proteins because some of our definitions of viruses is they need to make virions. So you have to have structural proteins. That's not terribly surprising. What the difference is with these viruses is, again, how your genome is actually going to get replicated. It is going to use all cellular proteins, but these are very, very funky genomes. And so that's what these non-structural proteins are about, is helping that genome replication to take place. So funky genomes, single molecules, I like to call them, rather than single-stranded. But they have a free 3' end. So again, they're not circular. This free 3' end is not just hanging out, flopping by itself. It's in the secondary structure, whereby you have a 3' OH and a template. Cellular polymerases love this, so they can go ahead and replicate their way through to the opposite end of the genome. The problem is, as far as the virus is concerned, is how do you now make more of this? You've made a double-stranded thing from what was single-stranded originally, but you want more single strands. So this is where those non-structural proteins come in. They make a nick in this double-stranded piece of DNA here, which again provides a 3' OH and a template. Cellular polymerases replicate out to the end. These structures can reform, and you have replication back out to the other end. These guys have helicase activity, but it's not sort of a replicative helicase activity. It's about pulling the two strands apart, excuse me, and allowing them to come back together. And some, again, well, we won't prep much for that, but some of the circoviruses and the cruciviruses, some of these have extra helicases, some of them don't. In the case of these parvoviruses, they all have helicases, but again, it just allows the formation of these secondary structures at the ends. So Nick basically provides a template for the polymerase to replicate from, and in some cases, helicases. This turns out to be almost identical to the rep proteins, also known as the non-structural proteins for the circoviruses. They do basically exactly the same thing. They have a double-stranded piece of DNA that they will nick, provide a 3' OH for the cellular polymerase, now replicating here in blue, to make a whole copy of that genome, in the process displacing this other strand. Most of these rep proteins, as I mentioned, have a helicase activity as well, which will separate this strand. Big difference here is that these guys have a OH on the protein that the other end is attached to. So the phosphate, which was here, that was attached to the OH here, which is now made by the cleavage, is now serving as the place to hang on to that extra strand of DNA until you get round to the other end of the genome. This will now religate, make another nick, and you can go through the whole process again. So it's a nicking process in double-stranded DNA. Helicase activity here now, that nicking activity is reversible, whereas in the case of the parvoviruses, it's not a reversible nicking activity. So why is this so cool? Because we found viruses that use this process probably to steal RNA from a co-infecting RNA virus. Totally bizarre, and it's actually talking to my daughter about this morning at breakfast. And so classification of things. How do you classify things? Humans love to classify things. But then you come up with this stuff that's there. Biology doesn't care about classification. And so that's where you come up with some of these really bizarre things, and then you have to try and come up with a way to explain them or think about classifying them. So originally we called them RNA-DNA hybrid viruses because this gene was stolen from an RNA virus. Then people didn't like that because it's actually really a DNA virus, not an RNA virus. So then we called them chimeric viruses for a while. Now we're calling them cruciviruses just because we want to have names for these things, and humans love names. It's not just crazy people in the Stedman lab who think this is happening. Other people have gotten on our bandwagon and found some more of these things. Whole set of these now chimeric viruses, and again, more recently called cruciviruses. And we're still trying very hard, particularly George and Nacho. So we got the two major researchers in cruciviruses gave lectures to you in this class. And well, we're still trying to figure out what is going on with these guys. So now the largest of these double-stranded DNA viruses, or DNA viruses in general, why do we do it this way? Because it turns out that their genomes are probably replicated in an extremely similar fashion to these parvoviruses. So smallpox, of course, how could we get rid of it? A great vaccine and no other hosts. Vaccinia is the way that we got rid of it. It wasn't cowpox. And of course, we had Antivaxxers, pro-diseasers, back in 1812 because clearly you're getting cows that grow out of your arms whenever these are grown. But believe it or not, and in fact, you could have had smallpox on this list as well before. Oh, here it is. And this was even in the US before vaccines, 30,000 people. After the vaccines, we now have none. And I particularly like this graphic because I think it really emphasizes what a great advantage these are. Measles had gone from 500,000 a year to around 100, almost all of these imported, and almost all of these among people who were not vaccinated. So vaccinate, vaccinate, vaccinate. If you get nothing else from this course again. But I won't give you 100 on the exam if you just write vaccinate, vaccinate, vaccinate on it. Sorry. So as I mentioned before, the replication of these genomes is very similar to what happens with the really tiny DNA virus genomes. You have these secondary structures that form at the ends of the genomes. Probably nicks. It's not entirely clear how these, the 3' OHs are generated here. Make cuts at this end of the genome, provide a 3' OH. Now, as I neglected to mention, but we've kind of talked about already, pox viruses and these other nucleoside large DNA viruses have practically all the machinery, definitely for DNA replication, that they need. So they've got their own polymerases, they've got their own helicases, they've got their own primases, etc. So it's pretty easy for them to make these whole copies of the genomes, but the structures, again, at the ends are such where they're these looped structures and probably are made as concatemers and then chopped up into smaller pieces later on. They don't have telomeres like our genomes do, and they're not open ends, they're covalently closed ends of the genomes. The virions of these pox viruses are, not surprisingly, large and complicated because we have many of these proteins that are also associated with, and again, why they call them tegument in herpesviruses and lateral bodies in pox viruses, no idea. That's just the way that they talk about them. But basically, the lateral bodies are the equivalent of tegument, or the tegument's the equivalent of lateral bodies. If someone were to ask you a question about that in an exam, that's how we would go about answering it. And what's in these are proteins that are important for very early aspects of the viral replication process. So, viral early transcription factors that you need for getting transcription of those very early genes. The RNA polymerase because it's got its own DNA-dependent RNA polymerase. That's also going to be present in these lateral bodies. So many of the enzymes that come in with the virion are actually present there. There'll be enzymes that are made as late genes because they're going to end up in the virions. The other thing about these smallpox viruses, and I think most other pox viruses, well, I'm not absolutely certain about this, is that some have single membranes, some have double membranes. The single membrane around the outside makes it a very stable particle, and so it can be spread easily from person to person in the case of smallpox, but also for vaccinia. It's actually a great vaccine because it can be stored dry, and it's actually literally called dry vax, where you can purify it. On the other hand, if you get an extra membrane, this is what allows the actin polymerization to happen to help move the virions from cell to cell. And that seems to be basically the only thing that that extra membrane is for. Mentioned already, it's this process where you have a virion enters the cell, has lots of viral proteins that are going to be involved in making first viral messenger RNA, then viral DNA. That viral DNA will end up getting packaged into these virions. Again, all of this in the cytoplasm, even though it's a DNA virus. These will get packaged now in virus factories that we didn't look at here, but we talked about quite a bit with the nuclear cytoplasmic large DNA viruses, and then enveloped in either 1, 2, or 3 membranes in order to get this released. 2 membranes when you end up releasing a single membrane form, 3 membranes if you end up releasing a double membrane form. As I mentioned, that extra membrane is mostly important for this cell-to-cell transport that happens due to remodeling of actin filaments. Those actin filaments will form behind a virion. Here's a virion. Here's the actin filaments which have formed behind it, basically sort of pushing that virion from one cell to another. Absolutely amazing process. How the heck that evolved, who knows, but again, an extremely fascinating process. So more questions on pox viruses before we talk about another kind of virion that packages a bunch of enzymes in its virion, which are these retroviruses. Retroviruses, RNA packaged in the virion, but DNA is critical for their replication process. All retroviruses have GAG, POL, and ENV. Oh, sorry, yeah.
Is it safe to say that the larger the virus is, less splicing involved?
Okay, so is it safe to say the larger the virus, the less splicing is involved? As far as I know, no viruses, and again, this is as far as I know, no viruses actually encode splicing machinery themselves. And so if you're going to be undergoing this process, say in the cytoplasm, like the pox viruses do, all the splicing machinery is going to be in the nucleus. So you're not going to have splicing take place. You can have, and we maybe get a chance to talk about this, in some of the nuclear cytoplasmic large DNA viruses, like the phycodNA viruses, those have self-splicing introns. So they're introns, but they can splice themselves out. So they don't require the splicing machinery. So you'll have some of that self-splicing, but in general, if you think about particularly things that are replicating in virus factories and are not using the nuclear machinery, you'll have less splicing in those cases. But again, it's always, there are always going to be the exceptions to these things. Okay, so retroviruses again, GAG, POL, and ENV. POL are really all of the enzymatic activities, protease, reverse transcriptase, and integrase. Envelope are the proteins that stick in the envelope, surface and transmembrane proteins. And then the other structural proteins, which are encoded down here at the 3' end of the genome, matrix, nucleocapsid, and capsid. The standard retrovirus infection cycle, interaction with receptor, fusion at the plasma membrane release of the capsid. In the capsid, with all those enzymes that are also being packaged in the capsid, you have reverse transcription that takes place. So you go from the 2 copies of your RNA to 1 copy of double-stranded DNA. This then has the integrase associated with it. That integrase protein, together with the double-stranded DNA, gets into the nucleus somehow. Most retroviruses actually wait until the nuclear membrane is degraded and then get in. In the case of HIV, you have the VPR, which allows that integron to get into the nucleus, even in cells that are not undergoing mitosis at the time. Once you have an integrated double-stranded form, this gets transcribed. That will get translated. 2 kinds of translation take place. You'll either have translation of a singly spliced version from this splice site to that splice site, which makes your envelope proteins, or a non-spliced version, which will make mostly structural proteins, but occasionally you'll have a frame shift that takes place in this messenger RNA, and you'll make a polyprotein. Of course, all of these polyproteins, just like those RNA viruses we talked about way back when in the course, those get chopped up by the viral protease into the smaller pieces that they need in order to assemble the virion. And then in the actual final virion, you have the last steps of proteolysis, which will form your final capsid and nucleocapsid structure. And those of you who haven't had a chance to look at the HIV video that I linked to earlier on, I think it's really useful in terms of the particularly assembly process and replication process of these viruses. The key to all of these retroviruses, of course, is the reverse transcription step, how you go from a single-stranded RNA to a double-stranded DNA. All happens through a cellular tRNA, so lots of different primers. We've got protein primed. We've got the priming that happens through cellular primases, and we have priming due to tRNAs. This tRNA primer, specific for the retroviruses, provides a 3'OH, base pairs with your viral genome, OH template, RNA template. It has to be now a reverse transcriptase. We'll make DNA. That DNA-RNA hybrid is now a template for the RNase H, which is part of the reverse transcriptase. That gets rid of the RNA. Now this DNA is looking for something to bind to. It can bind over here at this end of the same molecule or on the other molecule of RNA, which is present in the capsid. Now we have another nice 3'OH, a template that can replicate through the other end of the genome. Now we have a mostly hybrid, full-length now copy of the DNA and a shorter copy of the RNA. This gets broken down. It does turn out this is a polypurine tract, not a polypyrimidine tract. I looked that up last night. So the polypurine tract, I'll just call it the PPT. That's the last thing that gets degraded. This now has a 3'OH, a template. Reverse transcriptase can make a DNA copy of this through the primer binding site on the tRNA, gets to part of the tRNA that it can't recognize, one of the modified nucleotides in the tRNA, gives you a copy of the primer binding site here, which of course is complementary to the primer binding site over here. Second strand transfer, this short piece goes over to the end here. Could have been from that same piece of DNA or the other DNA, which is being made from that other RNA. Now you end up with double-stranded copy of your genome, including the long terminal repeats. So the unique sequences from the 3' end end up at the 5' end. The unique sequences from the 5' end end up at the 3' end. This is what gets integrated into the genome in a random fashion. Random, actually, probably not quite, because these have a tendency to integrate into actively transcribed genes because they don't have nucleosomes associated with them and they're not compacted. So integration happens. The integration process will cut offset in the DNA target site. It's not always GGG-CCC, but it always provides an offset cut, which means that you have a small gap that forms in the sequence after you have integration. That then gets filled in by the cellular repair machinery. So we end up with a short direct repeat of the cellular DNA on the outside, followed by the long terminal repeat, and then, of course, the viral genome between these two sequences. And there's actually a pretty good video for integration here, which talks about how those individual pieces are getting degraded. A little bit about HIV, slightly different structure, but the main difference here in terms of the structural proteins is this VPR, which allows the double-stranded DNA to get into the nucleus even in the absence of nuclear membrane breakdown. There are a couple of extra proteins, VIF, VPR, VPU, that we're really not going to talk that much about, again, other than this VPR, which is helping to get into the nucleus. VIF, VPU, and NEF, absolutely critical and important and fascinating, but they won't be on the exam. TAT and REV, on the other hand, are proteins which are important for transcriptional activation, but really anti-termination in terms of getting transcription to take place. So functions a lot like N and Q proteins, thinking about that other virus that we're forgetting about. N and Q from? N and Q from? Exactly. N and Q from lambda, so anti-termination proteins. There's a whole, it all makes sense, right? Or not, as the case may be. Serves as a very similar role for anti-termination of transcription, allowing you to get full-length RNA. TAT and REV, the REV protein is an RNA shuttle protein. Most important about the REV protein is the RRE site, the REV response element, which causes RNAs to get transported back out of the nucleus. What I absolutely spaced on mentioning when we first did this lecture, probably because I was getting close to the end, was that the REV binding site is right here where the REV protein is being made through this splicing. So the splicing that happens to make the REV protein is where this RRE site is. So the only RNAs that REV can take out of the nucleus are the ones that are not coding itself. They're coding for either the envelope proteins or the full-length RNA. And the real key about that full-length RNA, and we talked about splicing and blocking splicing before. Here splicing is happening, and usually if you don't have a spliced RNA, it sticks in the nucleus. But if you have an RNA genome that needs to get packaged, it's got to get somehow out of the nucleus. And so that's what REV is doing. So REV is binding that full-length RNA, transporting it out of the nucleus so it can get packaged. So again, that's what I spaced about mentioning the first time around. So we also have the giant viruses that we talked about. I didn't bring my double beta barrel structures. These are forming those icosahedral structures that you have in giant viruses, you have in PRD1, the bacterial virus, and also in STIV. Coccolithoviruses are some of the most abundant viruses on the planet. They make global plaques that you can literally see due to the lysis of these coccolithophores. This is the genome of mimivirus, but many of these other genomes, and this is sort of, yes, they're huge genomes as well as being huge virions, but they have more and more, the more that we sequence, the more that we find, translational machinery genes. So tRNAs, cap binding proteins, etc. The giant viruses escape, at least some of them, through these Stargate structures, which form on the virion and have that release. The difference with the archaeovirus VAPs is those are structures that form on the host surface. And then these megaviruses can get sick. There are virophages. These are infecting the virus factories. And basically, you can also think of these, if a permissive cell for a virophage is going to be one which is infected by one of these mimiviruses. So we didn't get a chance to review the most important ones, but they'll still be on the exam. People have more questions about those. Let me know. We can hang out here for a little while. Send me email. See you on Tuesday morning at 8. Good luck.